Chapter 6, Section 5, Transcription in Bacteria. Transcription, okay, you're transcribing, staying in the same language, just copying what was said, what was written. So here we're looking at RNA synthesis off of a DNA template. Transcription can yield a strand of RNA. That RNA can become a messenger RNA. That RNA can become transferred, ribosomal, or one of the regulatory RNAs. Remember, the difference here between the DNA and the RNA is the deoxyribose, or the fact that uracil U's instead of T's. For all intents and purposes, everything here is going to typically be single-stranded when it comes to these RNAs. They may fold. Once it's made is, a two-dimensional shape, just a string of RNA nucleotides, will fold in on itself in complementary base pair with itself, forming hairpin structures that'll give it a three-dimensional structure. So we're looking at RNA polymerase, specifically DNA-dependent RNA polymerases. This is different than what we talked about last chapter with viruses that have their own RNA-dependent RNA polymerase. Turns out bacteria and eukaryotic cells only have DNA-dependent RNA polymerases. Does the phosphodiester bond between the ribonucleotides? Similar mechanism to everything we talked about in the previous section. Can only add into an open 3', but it turns out RNA polymerase does not need a primer. Okay, it can just start building on its own. There's going to be certain sequences that are going to be transcription terminators. When it hits this string of nucleotides, the RNA polymerase will become unstable in its interaction with the DNA template, and literally it'll just fall off. End of. So it hits the termination sequence. Its interaction with the double helix will become unstable. The reason being, the hairpins and whatnot that the messenger RNA here will be forming will cause it to lose its interaction with the double helix. It starts to fall off. What it's showing here is that you can get multiple RNA polymerases moving down on the same one. As they're moving down, there's going to be more and more and more messenger RNA made. And what's kind of interesting we're talking about in the translation later in this chapter is that as the messenger RNA is still being made, ribosomes will hop on. So you'll end up having transcription translation occurring at the same time because there's no nuclear membrane separating transcription from translation. You have what is referred to as RNA polymerase holoenzyme. Okay, it's a collection of proteins and enzymes that their one and only job is to make an RNA transcript. You have a promoter. So you're going to have sigma proteins that are going to associate with the RNA polymerase. Going to help it, position itself at the beginning of the gene, beginning of the operon. It's this RNA polymerase that's actually synthesizing the RNA. You have other parts that are going to make an internal promoter sequence. Are going to interact with the promoter sequence, tells it where to go, when to go, and then when to start. When you look at the RNA polymerase across the living cells, bacteria, archaea, eukaryotes, there's going to be multiple portions, alpha, beta, beta prime. I forget what the Greek letter W thing is supposed to be. Keep wanting to say Wu-Tang, but that's a little old and dated. Archaea, a little more advanced, eukaryotes, quite a bit more advanced with what's there, what they can do, and what. Turns out that the promoter sequences that the RNA polymerase will interact with, there is quite a bit of conservative, sequences there. You'll have the start codon, the mRNA, where it needs to start. Upstream of it, you're going to have a TATA box, known as the Pribno box. And then at -35 upstream, so 35 nucleotides up, you're going to have another promoter region. These two promoter regions are where a lot of the initiation factors, a lot of the transcriptional activators will bind and interact. Sigma factors is a class of proteins. You'll see them Greek letter sigma and then a number designation. Okay, there's quite a few of them that have been found across the bacteria, and they just name them this way, sigma 70. The -10, the TATA box, also known as the Pribno box, and then -35 promoter, the -35 promoter. So this is -10 means it's 10 starts 10 nucleotides upstream before, so you hear upstream, downstream. Upstream means before the beginning of the gene. Downstream means after nucleotide number 1 of the gene itself. You're going to have strong promoters, weak promoters, and promoters that are just there. Strong ones are going to have a greater, the polymerases and whatnot are going to have a greater affinity for. There's going to be more sigma proteins, more transcription initiator proteins that want to bind there and recruit in RNA polymerase at a greater degree than many of the other genes that may be present. There's quite a few of these sigma factors that come into play here. Sigma 70, 54, 38, 32. You'll also see them referred to as RPOS, RPON, RPOD. They have different sequences that they recognize upstream of the gene, and they come into play at different points in time. Oxidative and osmotic stress, you'll see RPOS, also known as sigma factor, sigma 38, kick in, goes and binds the promoter region, this sequence, and initiates transcription of certain operons and regulons in response to one of these stress events. Heat shock, sigma 32 or RPOH, will go and bind once it gets activated because of a heat shock response. You're going to see things are going to be done in what are referred to as transcriptional units. It's the DNA segment that's going to give you one RNA molecule. You have initiation sequences and termination sequences before and after. You'll see this, if it's an operon, there will be 1, 2, 3, 4, however many co-transcribed genes. They're all put on one RNA, and they're referred to as polycistronic. You can also have transcriptional units where a bunch of co-transcribed genes, but they may not be exactly at the same place on the gene. Remember, those are regulons. They could be spread in different places. But the transcription factors that are going to initiate the first one are also going to bind the promoters at those other regions with the same affinity, and you're going to get a bunch of genes all at once. So let's see. You have a gene, a gene, a gene. Promoter, termination, transcription terminator. Promoter is going to bring in RNA polymerase. It's going to start right here, and it's just going to go all the way through, making one long RNA strand. The RNA, as it's being made, is going to go through and complementary base pair with itself, forming those stem loops of three-dimensional structures. And within the three-dimensional structures, they can either self-splice, or there'll be other enzymes that are going to come in and cut. The purple, that's just junk stuff. It's going to be degraded in the nucleotides, recycled. All the other stuff, well, here's 16S ribosomal RNA, there's the 20S. Here's a tRNA, one of them. Here's a 5S ribosomal RNA. All done at once, and then will be processed individually. If it's polycistronic, remember it's going to be an operon. You're going to have the promoter regions here. They're all the transcription factors are going to bind to, and they're going to recruit in RNA polymerase. They're going to recruit them in, all those transcription factors. First one binds, the next, and then all those together are going to bring in RNA polymerase, and then they're going to position it correctly so that it can start and then make one long messenger RNA. As it's made, boom, all three, they call them open reading frames. ORF1 is from gene 1, ORF2, gene 2. Well, then it's going to be spliced. I'm sorry, not spliced yet, because translation will occur and it'll be one long, polypeptide. Couldn't think of the word polypeptide, which will then be cut. Or ribosome binds, goes through, makes, hops, falls off. Different ways depending upon what it is. The termination, well, it's going to be DNA sequence based. It's going to be very GC sequence based. A bunch of inverted repeats. GG, CCC, repeat, CCC, GGG, things like that. It's going to cause stem loops to form, which are going to interfere with the activity of the RNA polymerase, which is going to make it slow down, begin to falter, and if it slows down and falters or comes to a stop, it just falls off the double helix. Transcription complete. So here's an example of the complementary base pairing. As the messenger, or as the RNA, it doesn't have to be messenger RNA, it could be ribosomal or tRNA, as it's being made, it's going to fold back in on itself because there's certain sequences. You see here, U, C, C, A, C, U, G. Well, hey, guess what? There it is, the same thing, and they can bind to each other. This is a stem loop. You get the stem loops, certain structures of them will destabilize the interaction of the RNA polymerase with the double helix. Destabilized, falls off, transcription done.